One is Nihesh Rathod. He's from Strand Life Sciences, and he's going to talk to us on reading between the lines, challenges in detecting methylation signatures in cell-free DNA. Nihesh.
Put in pocket.
Uh, Hello everyone, I am Nihesh from Strand Life Sciences, and today I will be talking about the challenges in detecting the methylation signature in cell-free DNA. Now let's start with the basics. Why would we study methylation in cell-free DNA? So during the development phase, different tissues will acquire very stable and unique methylation or demethylation patterns, and that would be like preserved lifelong. Now this study shows that using like what this study is showing is that we have 39 different tissue types, and from each of these 39 different tissue types they have selected 25 different regions that are showing unique methylation or demethylation pattern in each of these. So in total with 1000 regions you will be able to differentiate between these different tissue types. That is so if somebody wants to study DNA methylation, this paper is a really good paper to read. From the commercial perspective, there is one paper that came out somewhere in 2020. What they showed was that during using the DNA methylation pattern, they are able to detect the early signs of cancer, like much before the actual symptoms. Actually, 4 years before the symptoms, right? So after in recent years, what has happened is that lots of pharmas are actually putting a lot of money into cfDNA based multi-cancer detection scheme. So that's why we are really interested in studying this methylation in cfDNA. Now what is methylation? What happens is that in the genome you have certain places where C will be followed by G. These are called CPG locations, or where a methyl group gets binded to only the cytosine. Rest of the C's, if they are not followed by G or they are C followed by C's, they will not acquire any of this methylation, right? And as I said, this methylation regulates gene expression, and that can lead to different expressions of different genes. In the context of cancer, what would happen is that in normal cells, this methylation will be at certain level. For example, certain genes will be active, and they will be active because the promoters of those genes will be hypomethylated. That means that it can function very well. But in cancer, this behavior will be altered. What would happen is that the cancer inhibitor genes would get hypomethylated, because of which they are like the behavior gets changed, and it will lead to a lot of uncontrolled growth. Now, if you want to detect methylation, how would we actually detect it? Because the sequencer technology only has capabilities to identify A, C, G or T, right? So we need to do something extra to detect this methylation, because methylation is an epigenetic marker. So what we would do is that we would take this DNA from any sample, and before passing it to the sequencer, we have to use something called a bisulfite conversion, or it can be enzymatic conversion as well. What it will do is that wherever a CPG has a methylation, it will not alter it, but if there is a C that is not methylated, it will alter it to the uracil, and subsequently the PCR cycle will convert it into a T. Now when we do this kind of conversion, after the sequencer has done its job, we will get individual reads, and from these individual reads, when we try to actually map it back to the genome, we have to be careful, because we have already altered most of the C's, right? So we have to use specialized aligners to do mapping from individual reads to the actual reference genome, right? Now when we do this, there are multiple challenges that comes while detecting the actual signal. So the first one is signal to noise ratio. What would happen? So let's say that a doctor is suspecting that a patient has cancer, right? So what he would ideally do is that he would collect a tissue sample and send it to the lab. In the lab, when we try to get its sequence, it will have a very huge tumor or very huge cancerous signal if it's a actual cancer patient, right? And when we use this kind of sequencing to derive a biomarker, what we would see is that there is a strong signal that separates control versus a cancer patient, right? So this can act as a good predictor when we do biopsy. But since we are trying to detect the same signal from blood, from a cell-free DNA, what would happen is that a blood is a very heterogeneous medium. It has different kinds of cells. 55% would be blood cells, then 30% progenitors, 10% vascular endothelial cells. Those actual tumor signal, if it's present in the blood, it will be somewhere between 0.1% to 1%, right? So if we try to use the same technique at the same biomarker location, what would happen is that control and cancer becomes almost equal. Why? Because 99% of the signal is coming from normal cells, right? So now, is it still a good predictor? How would we verify? How would we use this kind of techniques? And what kind of techniques go into actually purifying the signal to a level where we can actually distinguish between control and cancer, right? So that's the first part. So let's go there. Now let's see this from the context of a sequencer. So when we send a control sample and do all the processing, it goes through the sequencing. If this is the region that we are interested in, there will be multiple reads that will be lining up here, and they will be showing some kind of methylation pattern. Similarly, a cancer profile will have a higher methylation, let's assume that, and then it will, so most of the reads will actually also show this kind of higher methylation pattern, right? Now, what we have done is that we have actually accumulated all of this signal into a single line here, accumulated all of these individual reads into a single line here. That's why it is showing the difference between cancer and control. In the case of cfDNA, what would happen is that the control will remain same, but in the case of cancer, most of the reads will be similar to what a healthy sample looks like. Only a few reads will be like this. So what we have to do is that we have to go ahead and actually, instead of processing it at this aggregate level, we have to look at the individual reads and then try to compare it with the healthy individuals, and then isolate all the reads that are not looking like a normal sample, right? So what we would do is that we will collect, let's say, 10 control samples, we would sequence them, and we will build a database of all the reads that could come from a specific region that is called, let's say, control read profiles. In the case of cancer, we would again do the same things, but then once we have these read profiles, we would go back and check here how many of these reads are actually looking like control samples, right? And after this comparison, we would end up with this red color reads which are not being seen by any of the control samples. That means that these are aberrant reads. That's what we call it. Those are called aberrant reads. And now, instead of using this aggregate signal for our classification, for building our model, what we would do is that we would actually count this. In a given region, how many of such reads are present? And based on that, we will try to derive the model. We will try to see whether we can identify cancer versus control. That's the idea. Now, how would it pan out in actual scenario? So this is a graph that shows false positive rates versus true positive rate. Now, when we use aggregate metric means, let's say a region has 10 CPGs. Out of them, if 90 CPGs are showing methylation, then it will count as a 1 signal. Otherwise, it will count as a 0 signal, right? So if we use that kind of aggregate matrix, this is the performance that we get. But at the same time, if we use this read-wise methylation, what we would do is that we'll build it from, let's say, high methylation percentage reads only. We will select only the reads that are showing 80% methylation or more. Then we would improve the performance of our model by quite a lot. Plus, what we have to understand is that we are actually not operating in this entire regime. We are operating only very near to this 0 regime. Why? Because we would like to minimize the false positive rates. We would not like to tell anyone who is a normal person that they have a cancer, right? Because they have to actually go through a lot to actually see that they don't have cancer, right? So we don't want to do that. That's why what we are interested in is performance here in this region, right? But what happens is that even when we do this, there is an even bigger challenge that we need to face, and that is called conversion efficiency. Why? Let me give an example. So this is the reference. What we are trying to do is that this is the reference genome. We are trying to find methylation signature only at this position where CPGs are there. And the way it works is that using bisulfite sequencing. So let's say if every one of these CPG is a methylated CPG, then the bisulfite conversion will not change them. It will remain as CPG. But what if the conversion is not working? What if the read that you are sequencing didn't go through that conversion at all? Then also you would receive the same signal. Then also you would see these CPGs as being CPGs. You will falsely identify it as a wrong signal. So it is not possible to determine the read with truly methylated CPGs with a read that is insufficiently converted, right? So again, this is from a single reference. Actually, PCR makes it even more complicated to compare them. And so what we need to do is that we have to actually not only rely on these CPG locations, but we have to rely on the Cs and Gs present in the reference genome. Now, Cs you can understand why I can rely on them, because after bisulfite sequencing, Cs gets converted to Ts. But what about these Gs? Why would we use Gs as well? So let me show you by an example why would that be the case. So this is the original fragment. We are interested in all the locations where C is present, whether it's on the top strand or a bottom strand, right? What would a poor conversion do? It will cause this C's not to change. It will still be C's because of the poor conversion. It didn't got changed to T's. In case of a good conversion, this gets changed to U's, and subsequently when we perform PCR step, this U's gets converted to T's, and from this top strand, this bottom strand will be generated through PCR step, right? Similarly, same thing will happen with the bottom strand as well. There are 5 U's that are 5 uracils that gets converted to T's, and through PCR, this another read gets generated, right? Now, so sequencer will actually output each one of them separately. You will actually end up with 6 reads. So let's see what happens to each of these 6 reads, right? So in the case of poor conversion, you would see that all the C's and G's in the reference have been intact, right? C's will be like C's, G's will be like G's. For the bottom strand, what would happen is that all the C's will get converted to G's, and all the C's from the bottom strand gets converted to G's as well, right? Because of PCR step. C's gets complemented by G's. So this is what will happen. But in the case of good conversion, there are actually 4 different scenarios that will play out. So let's start with the top strand and its PCR product. Let's see how it looks from the reference point of view. So if we compare it with the reference, all the C's in the reference have been converted to T's, G's remain as G's. From the bottom strand, what would happen is that all the C's will become A's and G's would become C's. Correct? Now, from the bottom strand, what would happen is that all the C's from the top strand will still be C's, but all the G's got converted to A's here. And the PCR product of the bottom strand, what would happen is that all the C's in the reference strand will be converted to G's, and all the G's will be converted to T's. Now, with this 6 observation, we could derive a conversion table like this. So if the conversion is not good, you would see some reads retain this behavior. All the C's will be C's, G's will be G's, or C's get converted to G's, or G's can converted to C's. In the case of good conversion, you would see these 4 scenarios play out. C's to T transfer, C's will become T's, or C's will, if C has become A's, G's will become C's. G's will become A's, or C to G and G to T. These are the only 6 things that can go. These are the only 6 things that can come out of a sequencer. If the conversion is binary, in a sense that either the entire read will be converted or entire read will not be converted. If there is some partial conversion that can also happen, then you will see a combination of these things. Now you have to go through read by read basis and derive a conversion, and then use only the reads for which you are very sure that they got converted correctly and they are showing the signs of methylation. So now let me show the result. This is very interesting. What has happened is that since we are working with only 1% of tumor DNA, anything that is less than 99% conversion efficiency will not be able to use it in a normal context. Right? If you don't do anything, we will not be able to use it because the actual signal will be drowned by all the impure reads that will be coming from the partial conversion. So because of which, what has happened is that we had built a classifier that would try to classify whether it's a cancer or a control. Now since this is a hypermethylated region, because of this partial conversion that is going on here, there will be many reads that will show the sign of methylation, because of which the classifier will say it's a cancer sample, which is wrong actually. Right? Now what we have done is that after getting this result, we'll pass it through the truth table kind of chart that I showed. We'll only select the reads that has good conversion efficiency, and this is the result that we'll get. Okay? So this is the interesting part. Even though the conversion efficiency is 96%, that means that 4% of the reads are not at all converted, then also we will be able to recover the signal. And not only recover it, we will be able to recover it with a very good confidence that this is actually a control signal. All these scores show that very strong correlation that it's actually a control sample, not a cancer sample. Right? So with this we have we have come to the end of the talk. These are the summary from these 2 challenges that we have. The first one is that methylation at read level is very important when it comes to dealing with any signal from blood. Then conversion efficiency we need to determine at a read level, not at the sample level. Because if we derive it at the sample level, our models can actually go wrong and they will classify a sample as cancer or control wrongly. And the last one is that even with good conversion efficiency, what we need to do is that we have to make it go through the entire pipeline. We have to check whether the conversion efficiency is intact, and then only we should use it for our downstream processing. These are the people who have helped me with learning all these concepts at Strand. I am not a biologist by profession, I am an engineer, but they help me with understanding all these things that go through sequencing steps and how PCR works and how it gets converted to different kinds of bases. So would like to thank them, and this is my contact details if somebody wants to contact me. Thank you.
Thank you, Nihesh. It was a very clear talk. Thank you very much. Questions?
So I have a question that you are working on data generated by your group or publicly available data?
No, these are entirely generated by our group.
I see. And only targeted to CPGs or whole genome?
No, it's neither. It's a very targeted sequencing, but methylated sequencing.
Okay, targeted methylation.
Yes.
Okay, thanks.
Can you just go to your last slide? I didn't get your point where you, no, not this one, the previous one. Yes, here when you are, classifying some of the groups to be control with respect to the, I think, the score you have given here.
Yes, here.
Yes, so can you just explain this thing once more? I just got confused
Okay.
on the last point.
So a model will output a score. It will output a score between 0 to 1. You will have a certain threshold. That threshold is mentioned here. So 0.2685. Anything greater than that score, you would classify as cancer. Anything less than that score, you would classify as a control, right? So earlier our score was 0.78, 0.5, 0.8, 0.8. And because our threshold is at 0.26, you would classify them as cancer because it's greater than that. After the processing, after only considering the reads that are good converted, we will be able to bring it down to this score. And now, because the threshold is at 0.26, you will be able to classify them as control, which is the actual case.
And how did you derive the threshold?
We build a model. That's an XGBoost based model. It's a random forest model. So yeah, that's the idea.
Because your inference completely depends on the threshold.
Yes.
So if that threshold is, incorrect, then
Yeah.
you'd get incorrect.
Yes, that is correct.
Okay.
So you did UMAP clustering also in this case?
Clustering?
UMAP clustering to see whether you are, based on this threshold, whether your test and control groups are separately getting clustered?
Okay, so what you are asking, okay, let me understand the question first. Like,
That's
so
an
you are saying,
unsupervised mode of
I
learning.
understand. What you are trying to say is that, let's say we are targeting 10,000 regions, we would observe values in them, go through UMAP clustering. Are we able to separate these two groups? The answer is that if we don't do this processing, you will not be able to do it.
You know, after processing also,
Yes.
say if I check it, as he was also asking you
Yeah.
whether regarding this threshold.
Yes.
So now you can have the two separated out totally.
Yes.
Okay.
Really nice talk. I was wondering if genetic variation can affect some of these results, right? Because you're assuming that there are no single nucleotide variants in any of these CPGs.
That is correct. So that's why we have a small leeway in how we derive this scores. What would happen is that in a given region, we would consider a read to be aberrant. Let's say if it's a hypomethylated region, means that the normals are showing no signs of methylation. Only if the read is showing very strong methylation, like close to 1, then only we'll consider it. That will not happen with a single change in the base, right? It will only happen in a true methylated state, right? That's the idea. Yeah.
Good. Any more questions? Oh, there are some here.
Great talk. I was just wondering, have you checked these prediction models and thresholding across various cancers, because it would vary there, and also some viral infections like hepatitis B virus. So does this thresholding or this model works across these different study systems?
We have tested it against different cancers. That works. We haven't tested it against different virus infections. So I would not know. Maybe it would work, but I have to check. But against different cancers, we have, and it works.
Hi, thank you so much for this informative talk. I have a question. I'm curious to know the motivation behind choosing cell-free DNA. Is it particularly important in cancer, or?
Yeah, so normally the way cancer is diagnosed is that, first of all, this is an early detection method. That means that people should be able to take it every year or every alternate year. Now, ideally, different cancers have different ways of identifying it. So for breast cancer, you would do mammogram. For something else, you would do biopsy and send the signals to testing laboratory, right? Nobody would do it every year unless they suspect that there is something wrong in their body. And by the time you would actually realize, you will actually see the symptoms, you would be in a later stages of cancer where the survival rate drastically decreases. The idea is that people should be able to take this test every year or every alternate year so that they can identify cancers in their early stages. And blood is a really good heterogeneous medium. It goes through the entire body, so it should accumulate all the signals that is there. It should be a good indicator. The only thing is that because the cell-free DNA has very little signal of cancer, less than 1%, we have to follow all these QC checks before we can give a diagnostic. That's the idea. That's why we use cell-free DNA.
Thank
Good.
you.
We'll take two more questions. Yeah, there are two more questions there. Let's.
You said it's an XGBoost classifier.
Yeah.
So it's a multi-class or
We
because
have-
there's only two classes here.
Yes,
You could have used XGBoost as a binary classification also.
that's what. So actually we have it across different cancers as well. So there are like multiple models. I am showing only the one of the model that is performing like this here, which is just classifying one cancer type versus one control. But since we are dealing with multiple cancers, we have multi-class classifier as well. That's there. Yeah.
Yeah, hi. Thank you for the very interesting talk. So I was just curious to know that could we use this kind of a method to actually predict cancer progression? Could we predict the different stages of cancer? Like
Yes.
If,
For some of the cancer types, yes.
okay.
For some others, you might not be able to do it because not only do we have the cancer information here, we'll have stage information as well. And we do see the change, even though it's a minute change because we are working with ideally less number of reads. We would sequence and then throw away 99% of the reads to do this, right? So you might not catch it. But across different stages, we have seen some drastic differences for some cancer type, not all. So that's.
Thank you.
Thank you, Nihesh. It was a very, very clear talk. Thank you
Thank
very
you.
much.
Yeah.
And I thank all of you for attending this session. Thank you.